All right, well, might as well get started. So thank you, everyone, for coming. I always have a bit more anxiety introducing other speakers to give their talks than I have giving my own talks. It's not that hard to give a research talk out of my back pocket, one that I've done 20 times, but I always feel like a little bit of anxiety introducing somebody else because I always feel like, a real drive to say something witty, concise, brief, funny, and so on. And that always gives me a little bit of anxiety. But this particular time, it's a little easier. So, because to take a page from David Letterman, I mean, this is true in this today that my next guest, really needs no introduction. Anyone know this show, David Letterman? I guess. If he was on the David Letterman show, he probably would need an introduction. But but in this community, I mean, like how many people in this? I'm going out on a limb here. How many people in this room have used or optical neurophysiology, do optical neurophysiology, or have used optical neurophysiological data in there? And how many of you have not used a probe made by Lauren Luger? I mean, I probably ever. So we have, as a community, our field has seen farther by standing on the shoulders of Lauren and his amazing reagents, which seem to come out on a regular, almost every year. GCamp, 11 or 12 or 13 or wherever we are now. So it's a pleasure to have him here and to learn about the new tools he has in his toolbox that should hopefully soon appear in our toolboxes, and where we might go from there. So Lauren.
So I have to admit, when you started talking about how you get nervous introducing people, I thought you were going to talk about my potty mouth. But I will try to do better today. Thank you guys for having me. I know a lot of you guys, and it's really fun to see everyone. That's my fake title. That's the real title. So, I don't have questions or agenda. My job is to come to people like you and you say, hey, these are the most important questions, and but we need something that does this, and so I try to make the widget that does that. And so, as always, let me know what you need. My talks are super informal, so please interrupt me. The last talk I gave here was a choose your own adventure talk. We can do that if you want. I am fully prepared to give a choose your own adventure talk, but I also made like a normal linear talk that I'll start with, and if you guys get bored or whatever, then we can go to a choose your own adventure talk. So this is the most important slide, is that everything is freely available. Stuff that's finished and well characterized goes to the usual places. Stuff that's, unfinished, and maybe we have like 4 versions of this and trying to figure out which one's the best for various applications. Those are also freely available, but I don't want to, plop them all on Addgene if a couple of them are going to turn out to be terrible and we're going to have to correct the confusion. So just email me, and if you email me, I will also send you the slides of this talk, the slides of every talk I've ever given, and any preprints that we don't have published yet. So I'll quickly run through, again, these are not my questions. This seems to be, what would help neuroscience? It would be able to, tools that would tell you where are things, what cells are doing things, what cells are connected to other cells doing things, what are the specific molecules involved in things, and probably won't get to this today, but some, really gory details of what's going on inside the cells. So I'll start with what cells are doing things. As Aravi said, I think you guys, so I made this, I'm going to go at a higher level and kind of just give you the executive summary of the new stuff that you probably want. and go a little light on the, so I'm not going to tell you why calcium imaging is important, for instance, because I figure that you guys know that. But if you want me to circle back, raise a question, raise your hand or whatever. So, for instance, I'm just going to tell you, there's JG Camp 7, right? I mean, so that's been available for a year now. We published it on bioRxiv last year. Finally came out in Nature Methods this year. The reasons you might want to switch to JG Camp 7. So, one thing that's become abundantly clear as we make tools is that, early days, there definitely are tools that like lift all boats. So like going from GCamp 2 to GCamp 3, there was no reason not to switch, right? I mean, it was just better in every respect. But then as technology gets, really mature, you start getting diminishing returns and you run into trade-offs in various things. And so the tools, the final versions of things become a bit more niche, So for instance, with JG Camp 7, there are now 4 flavors. There's S for sensitive if you really need to detect like 1 or 2 action potentials. There's fast, which is not really fast. It's just not horrifically slow, but I'm going to show you, actual fast things on the next couple of slides. And then, if all your action is in Neuropil, for instance, like the Drosophila people have always complained about GCamp. They're like, look, there's nothing, our cell bodies do nothing. It's all out in the processes, and, when your thing is dim, I can't even figure out where my processes are, so I'm looking in the wrong place. And, it needs to be brighter, or otherwise I have to co-express a red floor, and then that's a pain in the ass. So we have this bright, high baseline thing. Conversely, if you're, doing like wide field whole cortex imaging or, whole zebrafish brain, then this would be a problem, right? If the baseline's really high, then you have all this background pollution. There you really want something that's like at 0 and then goes to a very high amount. So that's C for contrast. So those are available, and now I'll tell you that you probably don't want to order any of those because we started attacking GCamp again, trying to finally actually make it fast. I mean, calcium is not a super fast signal. It's not voltage. It's not neurotransmission. So, I mean, there's a fundamental speed limit. But let me just tell you that GCamp, as it is currently instantiated, is far away from that speed limit. We think somewhere between a factor of 6 and 10 between what GCamp is telling you and what you can see from like calcium selective electrodes that tell you what the underlying transient is. So again, I'm just going to give you the high level version of this. These are all available. This is cultured hippocampal neurons, and this is a single action potential that we're eliciting with a field stimulator. And you can see, the good old trusty GCamp 6S, gives you really good signal and noise, but damn, it's slow. And then what I just told you is awesome if you want to do fast imaging, JGCamp 7F, it's a fair bit faster, so faster on rate, because action potentials are so fast, a faster on rate actually gives you much better SNR as well as better temporal resolution, right? Because most of the time the calcium was going up and down again before GCamp's like, oh, some calcium, I better detect that. And so you've missed it. So K-on is like the thing for sensors that you really need to maximize, both for signal and for being able to say there. So, but you can see we got an SNR boost from faster kinetics, but it's pretty bad. So what we did is we took GCamp, and pretty much the only part of the sensor that we already haven't bashed to pieces was this bit over here where the peptide interacts with calmodulin. And we had reason to believe, for reasons I will not bore you with, that this was could be the rate limiting step of GCamp kinetics. And so what we did is we took like 80 different peptides here and pasted them in, and the re-optimized linkers and all that. And let me show, and we've done 4 rounds, and we're doing 1 more, and let me show you how it went. So, round 0, when we just swapped in the peptide but didn't really optimize much else, we're like, some of the GCamps were dead. Some of them, it's like, ooh, maybe it's fast, but damn, it's dim, and the SNR is bad. So then we were like, oh, let's redesign the linkers, redesign the interface. Oops, wrong way. And so round 1 was like marginally better. Round 2, that was a single point mutant right at the interface between the GFP and the calmodulin, showing that there are really sensitive sweet spots for here. And we've always known that there's a lot at this interface. But so a single mutation, it's like, wow, now we're in business now. And then round 3, again, pretty marginal increase. Then round 4, that was I think that's like that single mutant right there. Now we're talking, So, it's got huge SNR relative to, even the best GCamp 7s. And we've increased both K-on and K-off, so rise and decay, by about a factor of 4. And we're currently doing round 5, really polishing this guy, but we think that we can do another factor of 2 faster than this. And then we start to probably start to run into that speed limit. But these are available, and round 5 should be screened in December, and so those will be available shortly. And then obviously we need to put these in vivo in a variety of preps and make sure that that holds up. But this is on the horizon. You can go ahead and get it. Also, we you get what you screen for, but sometimes you get what you didn't screen for. And so we got some stuff for free as well. So this is what I've just showed you, that we made some really fast things. That's what we were trying to do. We accidentally discovered that we made one that's much more sensitive. So this will be, if you need to give it a name, this will be GCamp 8S. And then another class that we discovered, so it's like whack-a-mole with all the properties that you need to optimize for a sensor. And one of the places where GCamps have historically been bad is what we call dynamic range. Not this dynamic range, but the like spike dynamic range. So you want something that's low, that's good in like low spiking, but also has differential signal in a high spiking regime. The existing GCamps haven't had that, but we found something that we're calling high dynamic range GCamp that now, from like 1 action potential all the way up to 160, is a pretty linear straight shot up. So, if you have like, neurons that undergo vast transitions in spike rates, and you want to be able to have signal across all those regimes, this one might be for you. These are available. So that's all I have for GCamps. Does anybody want to ask a GCamp question before I show you something else? Yeah. Oh, these are a lot of mutations. So yeah, no, we mutated probably 50 to 60 protein positions, screened thousands of mutations by themselves, and end up, everything is a combination of multiple things. So these will end up being, you know, probably like 15-fold mutants from GCamp 6 and 7. But you don't need to know that because I'm just going to FedEx you the DNA and you just PCR it and go. So, or we'll send you live virus. Yeah, something back there. Oh, Adam. Yeah. Sorry, my vision's not the best.
So the noise levels on these different traces look different. Can you say something about the absolute brightness of these?
Yeah, I mean, so specifically as to these, I think these just had lower N. But so for brightness, most of these are still exceptionally bright. You know, obviously the ones with the big signal change, I mean, you know, FSAT is pretty much as high as it can get. I mean, it's already 50% brighter than GFP. So, you know, we're probably not much more headroom there. But by making K on and K off so fast, that gives you more flexibility, and you can actually have a non-zero F naught and still get a great signal or noise. Yeah. Yeah. So we're going to try some of these as extracellular calcium sensors. You know, I mean, it's different concentration range out there, different pH redox environment, but you know, a lot of our GFP-based sensors have worked well outside. And I should say these also work well in the nucleus, so either as NLS or H2B fusions or whatever. So I think that these will be general purpose. For really high calcium organelles like the ER and the mitochondrion, there's a whole different set of variants that I'm not going to tell you about today. But if you guys want something with a super low affinity, like 100 micromolar or a millimolar, then email me and we can hook you up. Next, I figured I would tell you about neurotransmitter sensors. You know, trying to get at the details of, you know, what's making those cells active that you measure with GCamp. You know, piggybacking off the question that was just asked, you know, mostly when you say, hey, where are neurotransmitters? Where are not neuromodulators? You mean, where outside are they? And so in that instance, I'm going to tell you about a sensor that's going to be displayed on the membrane. But there are fascinating questions with where are transmitters and modulators and metabolites inside the cell. And so I'm going to show you a few vignettes of that as well. This is published, and I've talked about this here before. So this is the first one we made, glutamate, but I'm going to go through this pretty quickly just to, you know, just to give you a sense of what it's like to make one of these things and how you have to kick the tires to be convinced that, like, this is really a thing that's doing what we think it's doing. So, and again, if you're going to sense one neurotransmitter, it's going to be glutamate. So we started with glutamate. Bottom line is it works. You know, I'm not going to bore you with the years of making that right there. But, you know, that was 3 or 4 years to make that thing. But you guys probably don't want to hear about that. So, but then as soon as you put it, so this is extracellular glutamate sensor. This is now a mouse running on a linear treadmill. We're imaging in hind limb motor cortex, and we're injecting into layer 5 pyramids, and it's labeling the distal tufts and imaging in layer 1. So, you know, I'll just give you the, you know, the bare minimum, just the facts, ma'am. When the mouse is running, you get these events. They can, you know, different ROIs. You know, you have running selective ROIs, you have forward running selective ROIs, etc. So there's an interesting phenomenon. Something is occurring with the running, and it's different with different ROIs. But let's dig in. So importantly, the signal, whatever it is, whatever is blinking, occurs at things that are plausibly spines. They're the shape, size, rough distribution along the dendritic arbor of spines. So that's good. If you add TTX, the signal goes to 0, so that's good. As part of, so I didn't tell you that the basis for all these sensors, we like to use microbial proteins when possible because they're like 800 million years diverged from the stuff in the model organism. So it's pretty unlikely that they're going to tinker with the cell as directly, so there are these new, D-Lite and all these GPCR-based sensors, they're based on receptors that are expressed in your model organism. And, they may still have some G signaling, they may have arrestin signaling, all these, so when possible, we like to take it from something very distinct. And one facet of that is that, this is the sensor part, the binding protein is from E. coli, and E. coli cares about eating the glutamate in your gut that's left over from your half-digested burger, but it doesn't care about the drugs. And so, it's completely unresponsive to any other molecule that you might want to put on your preparation. Whereas like a GPCR, that cares about the drugs. That's why they're drugs, right? So, you can't use those in conjunction with those sensors. When you put on either a seizure-inducing agent or the, kind of mysterious fast-acting antidepressant ketamine, both of which are known to increase extracellular glutamate, the signal goes up. So it's like, yay, we've kicked the tires enough. This is definitely sensing glutamate and not something else. Another good feature of these microbial paraplasmic binding proteins so again, back to kinetics. The K-on and K-off of these things are extremely fast. So this is stop flow fluorescence on purified protein. And if you do the math, you see that, that shouldn't be negative. It's almost diffusion limited in that, it comes on really quickly, but K-off is also very fast. So it comes, the sensor turns green, but then the molecule leaves. So you're probably not going to buffer it too much. And this is now in SLICE, where we expressed the glue sniffer over the dendritic arbor. And this is using Kasper Podgorski's SLAP microscope that through optical trickery gives you 1 kilohertz large frame rate 2-photon imaging. And so you're doing simultaneous super fast imaging and 2-photon uncaging of glutamate at 2 spots along the dendritic arbor. And you can see that this is the eye glue sniffer signal after the spot of uncaged glutamate. And you can see that it locally spreads a couple, tens of microns within, 50 to 100 milliseconds. And that is consistent with, if you just do the diffusion calculation, then, so we think that it's fast enough that you can pick up, small molecule diffusing in real time. So these things are wicked fast. And then, if you have a green glutamate sniffer combined with a red calcium sensor, you can do 2-color, input-output imaging of what's coming into a cell and what it's computing and what's sending out. So, I'm going to say that's enough on glutamate because that's been out there a couple years, and I was going to start going to the next ones. Does anybody have any questions on glutamate before we do GABA? Okay, so if you're going to do a second neurotransmitter, it's going to be GABA. This one was harder. We had to find a soil bacterium to get this binding protein from. GABA, it turns out to be the predominant plant alarm pheromone for when it's being feasted upon by nematodes. And so plant roots, let out these GABA waves, and so soil bacteria learn to feast off these. So they have high affinity, high specificity GABA uptake systems. So we co-opted one of those, and again, this one was harder. This took 5 years. But then we have a GABA sensor. What can you do with a GABA sensor? So again, this just got published, so I'll just show you a single application. So this is now again back in Cortex. Our collaborators are inducing a local focal seizure by injection of pilocarpine, and so this we're going to ground truth in a bunch of different additional ways. So since this is a seizure model, we put in an EEG electrode. And so it'll be like, okay, now we have, another ground truth. Let's compare the fluorescent signal with the electrical signal and see what you see. So after you inject the pilocarpine, this has all been worked out over the last 30, 40 years. But there has been a surprising amount of controversy as to what molecules are actually carrying the waves of the seizure. GABA's been in vogue for maybe the last 10 years, but before that it was glutamate and calcium and some, more random molecules. But even then, a lot of the details were unknown. So we wanted to, verify in real time in a behaving mouse, with subcellular localization, is GABA carrying the at least a wave of the seizure propagation. And so for the aficionados in the audience, these are the interictal spikes of this is not the full-blown seizing period. These are the interictal spikes. But you can see that here is the fluorescent signal on top. Here's the EEG. And if you average those 5 trials, you see that there is very good correspondence between the electrical signal of seizure propagation and the fluorescent readout of extracellular, I don't want to say synaptic, we'll just say extracellular GABA during this time. And so I thought that was really pretty cool. Again, you got to keep doing experiments to convince yourself that you're not fooling yourself. So, seizures are known to introduce acidification of the extracellular space, for instance. And so you could say, well, you got a fluorescent protein out there, and it's these pH waves that are modulating the sensor instead of GABA binding. So I have a plan for that. So what we did is, so don't worry about these 3 right here. Again, tools just have different niche modes. These are different combinations of affinity, kinetics, and membrane targeting. But this one right here is a mutant that completely abolishes GABA binding, but still has the same pH sensitivity. And so you can see when you express that one in the mouse, there's absolutely no response during an interictal spike. So this really is GABA that the sensor is detecting. So that was pretty cool to say, like, yes, GABA is carrying at least a big component of the seizure wave. We could pick up the different temporal components of it, and you could dive into its, these cells, the advantages that imaging gives you. You'd be like, these cells are doing it, these are not, and try to tweeze apart a mechanism. Any questions on GABA before I tell you about the third most important neurotransmitter? Okay, so acetylcholine, also pretty hard to make for different reasons. This also took 4 or 5 years. We went back to the well to prototype it in vivo back in hind limb motor cortex of a mouse running on a treadmill, still imaging in layer 1. I probably don't need to remind you guys about cholinergic signaling. So most forebrain acetylcholine derives from the nucleus basalis. So we're going to use this to our advantage when we prototype this sensor. So first, again, just like we saw with glutamate, when the mouse is resting, again, this was consistent with literature. At rest, the cholinergic signal sensor, gave a lot more crackling than the glutamatergic, the glutamate sensor, which is consistent with, in this circuit, there's a decent amount of tonic choline release. But when it runs, the spikes, the events increase in frequency. The label fell off this. What that is the addition of a acetylcholinesterase inhibitor. So, acetylcholine, most neurotransmitters are taken up by transporters like glutamate and GABA. Acetylcholine is instead degraded. And so you have this enzyme that if you cripple that enzyme, then acetylcholine hangs around for longer. And so these events became more common, and the baseline crackling, you could just say, hey, that's noise from low end. We've done this enough to convince ourselves that there really is more of this crackling occurring in the presence of the acetylcholinesterase inhibitor. And then, again, we made a single mutant that knocks out acetylcholine binding, and comfortingly, you get flat lines with no events, but also much less crackling. And again, I'm not going to show you the data, but we've done enough in that we think that this is a real observation that, maybe these are tonic or small acetylcholine release events that we're picking up there, but obviously not with the control sensor. So much less is known about cholinergic signaling than, for instance, glutamatergic signaling. I said for glutamate, first thing we did is we figured out if it spines. There's nothing like a cholinergic spine, but there's a fair bit of controversy as to what these postsynaptic structures look like. We did the same analysis as with the glutamate sensor and found that we were getting these hot spots. I don't want to call them spines, I'm just going to call them hot spots of cholinergic signaling along the dendrite. And in a different preparation, this is now a slice where this is clearly a soma, and you're having a cholinergic axon come along and innervate the soma. And our collaborator did some STED-like super resolution functional imaging to super localize the hotspots, and so these are the somatic release sites, and you can pretty much count them and estimate their distances and where they are in the soma. That was fun. Something else that's controversial about choline and I would say neuromodulators at large is we're always taught that there's phasic and volume signaling, and they have different modes. Some people say like choline is only volume. It's not a phasic neurotransmitter. This sort of argued to me that no, there really is phasic cholinergic neurotransmission. Something that drove the point home to me even more is, so this is now Casper's, again, the SLAP microscope that lets you do 1 kilohertz 2-photon frame rate imaging. And so what he's doing is he's imaging in layer 1 in visual cortex, but this is not behaviorally evoked. This is direct current injection into the nucleus basalis. I think he just does like a 1 to 2 millisecond burst of current injection, and so you get the shower of acetylcholine onto those dendrites. And by imaging so quickly, he can see that the acetylcholine seems to spread 1 to 2 microns, and the event lasts for 10 or 20 milliseconds. So to me, that's phasic acetylcholine signaling. This is not volume by any stretch. And then the last bit of data I'm going to show you about acetylcholine. This was fun. So I don't need to tell you guys that it's not clear how anesthetics work. That's actually wildly controversial. But one thing that we found, but by accident, the mouse was being anesthetized to do something like check the brain window. I mean, it was a super boring reason. But during the fairly brief period of medium isoflurane anesthetization, so a couple things become obvious. So you can see that the shutdown in cholinergic signaling is extremely rapid. That came on within 2 seconds of starting the isoflurane flow. Another thing that's a bit more subtle is that the effect was more profound at the deeper layers, and that it came on faster there. So it initiated faster in the deeper layers, went further along, and then recovered slower in the deeper layers. And I should tell you here, instead of, so this is still the SLAP microscopy, but instead of using the light to do super fast temporal resolution, he used it to do volumetric resolution through the top 5 layers of the cortex. This was not really known, the differential effects of anesthetics on cholinergic tone in the cortex, and starting to run short on time. So I have some theories. I think it has to do with levels of chat and acetylcholinesterase expression, but I won't bore you with that right now. Something that I didn't have time to show you data about is that using a Cree-dependent expression of the acetylcholine sniffer, we put it in pyramidal cells, somatostatin-positive interneurons, and VIP-positive interneurons, and the patterns of innervation by the cholinergic axons rising from the basal forebrain was different, and the distribution of hotspots looked different. So we think that the sensor can help get at the combinatorial code of cholinergic signaling. Any questions on acetylcholine before I show you our hardest one yet? Okay, show you serotonin. So the reason this was so hard is that serotonin is not an environmentally abundant molecule. If you guys have ever worked with serotonin or dopamine or norepinephrine and you make a solution of it and put it on your bench and you go eat a cup of coffee, you come back and it's this black tar, you know. So, I mean, you know, our freckles are essentially polydopamine. And so, you know, it's, you know, so I don't think that bacteria see this. And even within eukaryotic cells, they treat the monoamines with kid gloves. They keep them right near the membrane at all times. So almost every monoamine binding protein turns out to be an integral membrane protein. You don't let it just kick around the cytoplasm. So we couldn't just say, nature, give me a serotonin binding protein, I'll turn it into a serotonin sensor. So we're like, well, what the hell are we gonna do? Then we realized that, you know, serotonin and the acetylcholine that I just told you about, they have, you know, they're pretty different molecules, but at least they both have an amine. And so we did many rounds of computational design and machine learning and just throwing the book at it, and until we ended up with a not great but tolerable serotonin sensor. So, you know, it's got really good signal and noise, you know, 8-fold, 10-fold even we find in some preps. This should scare you. That's a pretty high, low affinity. That's weak serotonin binding. And, you know, if serotonin is kicking around at very low concentrations, is this going to be able to detect it? But what the hell, it's the best we had, so we forged forward. This is in collaboration with Viviana Gradinaru. You know, so there's a couple circuits where serotonin's role is fairly well established, and it's like, let's go there first before we go, you know, kicking around in some random region where it's super controversial because we won't know if we're right. But so they injected it into basolateral amygdala, and now there are 2 electrophysiological ground truthings. There's an EEG array to measure you know, activity, and then there's actual an EMG electrode to you know, measure more behavioral output. And so what we're going to look at is we're going to look in a, you know, a very simple system, which is sleep-wake, you know, where serotonin's role is well established. And so this is a busy slide. What you have down here, this is an ethogram of what the mouse is doing between wake, non-REM, and REM sleep. So this is, we don't have cameras on the eyes. This is just defined by, you know, low resolution videography on the mouse. This is the serotonin. This is the fluorescent trace you're seeing here. This is the EMG of what the mouse is apparently doing with its arms and legs and what have you. And this is an EEG spectrogram of what's going on around the amygdala. And so you can see that there's pretty good correspondence. You know, obviously when the mouse is awake, it's doing a lot of things with its muscles. So you get a big EMG signal. That is expected power for EEG of a waking mouse. And then you can, if you look closer, you see that at these various sleep state transitions, you get these little bumps. And zooming in on one of those transitions. So this was the biggest change that we saw between the three possible state change combinations. The biggest signal in with serotonin that we got was between REM sleep and waking. This is broadly consistent with literature reports with like microdialysis or fast scan cyclic voltammetry of what's going on with serotonin at these sleep state transitions. So I promised you a little insight into using the sensors expressed inside cells, which again, so that's another thing that you can't do with the, there are GPCR-based serotonin sensors, but their properties are not great. SNR is bad, they're really slow, but for the application I'm about to show you, they are just unusable because you can't express them in a way where you could look at intracellular serotonin. So what we've done here is we've expressed the soluble version of the serotonin sniffer inside cells, and we have overexpressed the human serotonin transporter in this cell. And then what we're doing is we're, again, for boring reasons, the way to get the most high resolution data is to oscillate the serotonin, ramp your buffer between high and low, rather than keeping it clamped at individual concentrations. And so, you know, when extracellular serotonin is high, we run it inside. When it's low, we run it back outside. And that's what you're seeing here in these standing waves. And then we toss on 1 micromolar of Lexapro. And comfortingly, you know, within, I think this is like 10 seconds, it totally shuts down transport through CERT. So, you know, Lexapro is at least in part working like we think it does. A more fun experiment was we looked at X, MDMA, effect on serotonin. This Lexapro is a competitive inhibitor. MDMA is actually a competitive substrate. So again, these oscillations were just verifying the ionic dependence of the human serotonin transporter, that it was indeed sodium dependent. And then when you apply, this is the period of application of MDMA, it competes with the serotonin to go inside the cell. And so you end up with a lot of MDMA inside the cell and less serotonin. And so you can see that you know, it's telling you there's less serotonin. When you get rid of it, okay, I think those labels are switched. Okay, any questions on the serotonin sniffer? And now I might need a little input on what you guys want to hear about for the next 10 minutes. Yeah, question? Yeah, so yeah, we have a negative control serotonin sensor, and it was flat. So yeah, it's not a pH artifact at least.
No, I think I just, from personal experience, you can see these sorts of 10% baseline shifts
Yep.
and changes during sleep.
Oh, yep. Okay, so there's a couple things I could tell you about. I could tell you about figuring out where drugs are in the body. I can tell you about viral engineering, or I can tell you about EM. Anyone have a strong? Sorry, what? Okay, viral. Okay, all right. I'm gonna tell you about viral, and then yeah, and then we'll see if I have time to show you one more story. Yeah, so this falls under the rubric of what cells are connected to what other cells. You know, there are some tools for this, but they historically have not been very good. This is I got to explain this a little bit. So what this is this is a Cre-dependent mouse that when Cre is present expresses nuclear localized GFP. So you're going to get green nuclei when Cre is present. The manipulation is that the virus is being injected into the basal pontine nucleus, and we're hoping to backlabel layer 5 of cortex. The reason we picked this prep for multiple reasons. This is a long range projection. This is, you know, a couple centimeters across the mouse brain, and this is also one of the largest projections in the brain. And so the notion is, hey, wouldn't it be great to figure out which cells in layer 5 are projecting to the BPN? And so what we've done, and so we have an AAV serotype, we're packaging Cre recombinase, we inject it there, and we see what we get. So we started with AAV2. So you'll find, in some cases, high-profile papers saying that like existing AAV serotypes are actually really good at retrograde transport. You'll actually see some that say that they're really good at transsynaptic crossing. I'll just say that I don't think that any of those results are remotely true. But I report, you decide. So this is AAV2, you know, a workhorse serotype. We inject it in the BPN, and we get generously 8 cells labeled in layer 5. So this was a collaboration with Dave Schaefer at Berkeley. We actually, for reasons that are lost to the sands of time, we did the actual selection in 2 different circuits. We went from one labeling striatal structures and one in the hindbrain between the cerebellum and the inferior olive. We had a capsid library of AAVs, and what we would do is we would inject in the target, and so each virus is packaging its own genome rather than a payload. And so what you do is you inject, and then like 2 weeks later you dissect out this part here, and then PCR up what you had, sequence it, make a second round of libraries, and then iterate. So after 5 rounds of selection, both from the substantia nigra and from the inferior olive, Fascinatingly, both libraries converged on the same motif out of a large initial diversity. And as a protein guy, what's really pleasing is that this is definitely amino acid dependent and not DNA dependent, because you get the same motif but in two different registers. And there was also, different codon usage, etc. So we're like, wow, this is doing much better. And now when you inject in the BPN, we labeled, almost 100% of layer 5. And we also, so some of these were unknown connections before this experiment that we have since followed up with injecting and doing anterograde tracing back down to the BPN. And just to quantify it, the thing I showed you at first, the AAV2 is terrible. These, both of these have high-profile Nature papers saying that these are wildly successful retrograde tracer viruses. They aren't. There's a different virus called canine adenovirus. This is a tricky virus. It's toxic, and it's also only produced by one lab in the world, and they're kind of cantankerous. But this has been the gold standard for retrograde tracing. You can see it's not good. Surprisingly, DJ, which was involved just to be to spread better in the cortex, decreased heparin binding, was good. This is the absolute gold standard, Fluorogold. This is a small molecule tracer. We back-labeled actually more neurons than with Fluorogold. And so, so we have a really good retrograde tracer virus in conjunction with probes that I didn't have time to tell you about. We took AAV retro and packaged it with a bunch of, they're called spaghetti monster antigens. We packaged a bunch of different flavors of the spaghetti monster antigens, and then did injections into like thalamus, pons, superior colliculus, and you can start to get, really pretty maps of brain connectivity. What time is it? Should I stop or should I? Oh, okay. I'll tell you a little more then. So this is a pretty picture, But if you want to do real science, so this was a big collaboration between us, Carell, a couple other people at Janelia, and importantly the Allen Institute. So the Allen, they just had their big opus on, here are single cell transcriptomes of a bunch of parts of the brains, these are the different cell types. So they, this is the result of, I think this is 30,000 cells with single cell RNA-seq, and this is a t-SNE clustering of the expression profiles. Each dot is a single cell, and then they're grouped by which cortical layer they came from. So I'm going to tell you about this little cluster over here, the pyramidal tract. Do people know what the pyramidal tract is? Okay, you guys seem to know what the pyramidal tract is. It's super important. So the reason that Allen came to us was that they couldn't isolate these cells. When there's something about the pyramidal tract neurons that when they were doing the microdissection and the cell sorting, these cells all died on them. And so they're like, look, we have nothing for this. And so we're like, oh, well, let's back label it with AAV retro to from areas that I'll show you that are projected to from layer 5 PT neurons. And so this was our contribution to get these cells labeled. You can see that they roughly cluster into these 3 different expression clusters. A big thing going on at Janelia is called the Mouse Light Project. I don't know if you guys have heard about that. What this is very sparse expression in cells in the brain, and then reconstruction of their full axonal projections. And so what you're seeing here is that these, I think this is 30 cells that were reconstructed by the Mouse Light Project. You know, the soma is right here, and this is the dendritic arbor, and this is all axon. So I mean, you can see this is technically a hard project. You have to do a lot of serial section, block phase 2 photon imaging, and then a lot of reconstruction because in addition to being so long, axons are maddeningly thin, and they're easy to lose. All this should tell you is that from the single cell reconstructions, we got an idea of the different populations of these PT neurons, that some would go to thalamus and some would go mostly to medulla. And there are a couple areas like pons and superior colliculus where they both went. And this backed up, just corroborated, what we knew from our retrograde tracing experiments, just saying we're not missing anything. These are what the cells look like. And so the 3 major areas that these pyramidal tract neurons pass through on their way to the brainstem are thalamus, medulla, superior colliculus, and pons. And you can see that by the projection experiments, it seems that there are different populations of these things. That the thalamus projecting neurons are superficial, the medulla projection neurons are deep, everything goes to the colliculus, and pons is a bit more weighted towards the deeper medulla projecting neurons. But then what was really cool is we then used AAV retro to back label the thalamus and medulla projecting cells, independently isolated them, did the single cell RNA-seq, and they map onto the one-to-one onto the expression clusters. So, the deep medulla projecting cells are of one expression phenotype, and there are, 2, maybe 3 subclusters of these superficial thalamus projecting cells. That was our contribution to the paper. Carell's lab did a lot, and so they were doing electrophysiological recordings from these neurons, and also channel rhodopsin stimulation experiments. And what they figured out is that these cells are the ones, so, motion planning is hard, right? You need to, you need to decide that it's time to go. You need to initiate the movement, and then you need to get proprioceptive feedback to see how the movement is doing and whether you need to fix it. And so through the optogenetics experiments, Carl was able to figure out that these guys are the one that initiate the movement, and these guys are the one that manage the proprioceptive integration of how long it's going to last, whether it's going to be successful. And then I'll just leave you with the notion that, you know, PT cells are implicated in numerous diseases. And so with Alan, we're, you know, obviously we can't do the AAV retro labeling in humans to get those populations, but they're doing the single cell on human PT neurons and see if we get the same or similar clusters to see if this holds in us as well. Should I stop there? Okay, I'll stop there. So, yeah, so again, you can email me for slides, some highlights of things you guys missed. Was the drug sensors, the labels for correlative light and good ultrastructure EM, and a bunch of other things that I didn't even bother putting in my slide deck. But if you want them, I'll send you slides on them. Does anybody have any questions? Okay. Oh, okay. Wait. You went first. Yeah. 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 I mean, that's on our list. Neuropeptides are going to be hard for a million reasons. So one, the actual phrase neuropeptide is kind of a misnomer. There's only a couple of them that you would call actual peptides. Most of them are neurosmall proteins. The other complicating factor is, you know, we know of, I think, about 200 different neuropeptides. I bet there's about 600 we don't know of. And a lot of them are, you know, like just single mutations away from each other. And there's an emerging consensus that neuropeptide receptors are probably somewhere on the axis between, like, you know, an AMPA receptor is, you know, it's just glutamate. But again, you know, it's a slippery slope, because then if you talk NMDA receptor, well, then it's like, well, you got the glycine and deuterium, you know, and there's probably other allosteric sites we don't know of. But for neuropeptide signaling, I think it's more of a combinatorial code akin to like olfactory receptors of, you know, hey, you know, we've got all these receptors, there's even splice forms and whatever, and it might be some sort of code between activity at all these different receptor types to compute like how much of that was in PI and how much was in enkephalin and all those sorts of things. From a scaffold point of view, I mean, yeah, you could use the GPCRs. I mean, I think, honestly, I think that's the only easy way to start. Those are kind of messy. And again, I always worry about like overexpressing a GPCR, even if you knock out the G protein binding. You know, these things do other things in the membrane that I worry you're going to make your cell crazy. There are bacterial binding proteins for peptides. They tend not to be super specific because, again, bacteria don't care which peptide they're eating. They just need amino acids. And so they've been designed to be less selective. I think we can design some, but that's going to be a long game, you know. And then figuring out what it all means, you know, I think. So that's a definite long-term goal. But that's the final frontier, right? We know nothing about neuropeptide signaling at this point.
My question is about forward-hopping proteins. Are you guys working on those? Are you taking a mutagenesis approach to that? And there's
Well, yeah, I mean, we're working on it by taking a mutagenesis approach to it. don't have anything to show yet. I think it's a big ask of AAV. You know, AAV was not meant to hop synapses. And any of these reports that you see that like, no, AAV9 actually, you know, crosses synapses, there, you know, there's a big difference between transneuronal and transsynaptic. And I think when a lot of people say transsynaptic, they mean transneuronal. Like, you know, viruses just get shed. You know, the cell's like, nope, exocytoses it, and it could bleb over to a nearby neuron. You know, there is precious little validation, except in cases of like, you know, a true transsynaptic virus like rabies. But that, again, I mean, that comes with its own caveats. It only backlabels 9% of synapses, and which ones? You know, we don't know. But that one's going to be a big ask.
Do you know what the signal peptide does that you identified with this AAV, like the little protein signals?
We do not, and that keeps me up at night. So I've talked to people, you know, I've talked to world experts, and I've got a short list of things I think it might be binding to. So, you know, axons are so long and so thin that, you know, it's like age of the universe to like just diffuse from, you know, the terminal to the soma. So it has to be active transport. You know, people's best guess is that, you know, it's hijacking the, you know, the kinesin motor system. So you get these, so AAV is internalized in endosomes. You get these like early, medium, and late stage endosomes. They change their surface protein profile as they move through the cell. I want to say it's, I think it's RAB5 is the marker for the medium stage endosome that we think might be grafting onto the kinesin motor system. And, you know, if I had to bet $1, I think we've made a binding site for one of these so-called cargo adapter proteins, and I think there's a chance that it's either hook 1 or hook 3. Yeah, I mean, yeah, we're gonna do, yeah, we're gonna, you know, for these small candidate gene lists, we can do experiments. You know, we'll just, you know, purify some hook 1 and just do BioCore and see, like, do they bind? We're doing, like, you know, scanning mutagenesis on that peptide to see, like, what are the critical residues, see if we can, you know, guess. Yeah, that one is pretty crazy. So something I didn't tell you guys is that AAV retro is super cell type dependent. It works well in glutamatergic and GABAergic projections. It doesn't work in dopaminergic projections, and it doesn't seem like it works in peptidergic projections yet. And so, I mean, it was crazy. The day the paper came out online, Twitter exploded, and like the first 100 messages were like, "Oh, we're going to cure Parkinson's," and then like the 101st tweet was like, "No, read this up. It doesn't label dopaminergic neurons." And then it was just like hate for like you know. So, but we have started tinkering both with the motif and also the serotype background. Like that was built on AAV2. We actually grafted it onto AAV9 and also made some other tinkering. it's preliminary, but it really looks like we're back-labeling populations of neurons that we didn't label with AAV Retro 1.0. But again, it's super preliminary, and we haven't characterized exactly what those cells are. But for instance, you know, it wasn't labeling AAV Retro 1.0 doesn't back-label layer 6 from thalamus, and that's bad. So, but we have a new mutant that seems to do that. But again, you know, it's a trade-off between like, you know, trying to dig in and understand the failure modes versus like, just keep your head down and plunge ahead, because you only, you know, the good thing about design is you only need one, right? You know, if I'm just walking along and I pick up a tube of DNA and I'm like, let's try this, and it works, you know, if I don't understand how it works, that's secondary to helping people. So. Yeah, oh, yeah, anything. Yeah, that's my job.